Hello, my name is Tim Camp. I'm visiting from the University of Wisconsin, and it's my pleasure to moderate this session entitled The State of Cardiovascular Regenerative Medicine. I'm co-moderating it with John Cooke, who is our first speaker. John is from the Houston Methodist Hospital, and he will present a talk entitled A Novel Epigenetic Process in Nuclear Reprogramming. John, please.
Thanks so much, Tim. Welcome, everyone, to this wonderful meeting that Jay has put together with Joel and GQ. I've been looking forward to this for quite a long time to hear what you all have to say. I know I'm going to learn a lot today. We have a very full schedule, so each of the speakers has 15 minutes. We have 5 speakers in this session, and Tim and I will try to keep it on time. So toward that end, I'll go ahead and get started without any further remarks on my talk, A Novel Epigenetic Process in Nuclear Reprogramming. Now, we've been working on this observation for a few years now that nuclear reprogramming, whether it's nuclear reprogramming to pluripotency or nuclear reprogramming for transdifferentiation or for merely a change in cell phenotype, that type of reprogramming to change the phenotype of a cell does involve innate immune signaling. So I think we're all aware of this pathway that's mediated by pattern recognition receptors. Pattern recognition receptors like toll-like receptor family or RIG1 or receptor against advanced glycosylation end products. Those receptors on the surface of our cells, they are there to recognize damage-associated molecular patterns or pathogen-associated molecular patterns. Those represent challenges that the cell needs to respond to. And the cell responds to that, as you know, through a complex signaling pathway that leads ultimately to the activation of transcriptional factors like NF-kappa B, or IF3, or IF7. And you know that that leads to the release of inflammatory cytokines. Some of you who've heard this talk before also know that we've discovered another limb of that innate immune pathway, and that is global changes in epigenetic modifiers, changes in their expression and activity that result in an open chromatin state. So today I'm going to give a progress report on the work that we're doing in this area, some old work for those of you who haven't seen it before, and some new data for those of you that are aware of the story. So essentially what I'm going to show you is that innate immune signaling causes changes in histone markings such as H3K4 trimethylation, an increase in H3K4, a reduction in H3K27, and other changes in the histone proteins that leads to more of an open state. So in a fibroblast, for example, the genes encoding pluripotency factors, the pluripotency cascade, those areas are in closed chromatin conformation. In an iPSC, of course, those areas are active and actively transcribing. So to go from a closed conformation to an open conformation takes more than transcription factors. You need to have epigenetic changes that permit that process to take place. And we showed some time ago in a Cell paper published in 2012 that the induction of pluripotency requires the Yamanaka factors, but it also requires innate immune activation signaling that results in an opening of the chromatin. Every technique that we use to generate reprogramming involves this process. What we've learned more recently, we haven't published this yet, is that the process, it has a Goldilocks zone. So there's an optimal range of innate immune activation for reprogramming to occur. The model that first revealed this to us was the DOX-inducible murine embryonic fibroblast model that you see here. You know, these are murine embryonic fibroblasts that contain a Yamanaka cassette, and with addition of DOX, within about 3 weeks you get these nice iPSC colonies, in this case staining with alkaline phosphatase. we found that as we increased activation of innate immunity, we could get more colonies up to a point when further increases in innate immune activation actually caused a drop in the yield of colonies, as shown by this red line. We had an NF-kappa B reporter construct, a luciferase reporter, that allowed us to see that as innate immune activation is increasing, there's an increasing yield in this range called the Goldilocks zone, but beyond that, things dropped off, yield dropped off. In addition, blocking innate immunity, in this case with a P65 decoy, resulted in a reduction in colony formation as well. Now, we've developed further evidence of this Goldilocks zone using an assay called a micrococcal nuclease assay. There's a number of assays to look at DNA accessibility. This is new data. In this micrococcal nuclease assay, the chromatin is chopped up into nucleosomal fragments. If there's greater DNA accessibility, you get more mononucleosomes versus trinucleosomes. So we again use this DOX-inducible MEF model, and again we explored the Goldilocks zone by activating the cells, the fibroblasts, with doxycycline, and then increasing doses of poly-IC, which is polyinosinic cytidyllic acid, a TLR3 agonist. And here's some of the raw data. This is what the data looks like from the bioanalyzer using a chip that's more sensitive to smaller DNA fragments. And basically what you can see is a peak that represents mononucleosomes, dinucleosomes, and trinucleosomes. And one of the things we did was to generate a ratio of mono to tri. The more mononucleosomes you have, the greater DNA accessibility. And you can see in this Goldilocks zone that I was showing you before that you have more mononucleosomes, as shown by this ratio in the black bars. The mononucleosome to trinucleosome M to T ratio is greater in the Goldilocks zone, meaning there's greater DNA accessibility in the Goldilocks zone. On either side of the Goldilocks zone, insufficient activation of innate immunity or too much activation of innate immunity, you have more of a closed conformation. Now, this phenomenon applies to transdifferentiation as well as the induction of pluripotency. So I'm an endothelial biologist. We wanted to see if we could transdifferentiate fibroblasts into induced endothelial cells. Some of you are aware of this work that we've done where we just, instead of using transcriptional factors, we simply added an activator of innate immunity to the fibroblasts, and we also put the cells in their state of epigenetic plasticity. We applied endothelial cell transdifferentiation factors, and what I mean by that are things that endothelial cells like VEGF and FGF and BMP4, and an antagonist against the TGF-beta receptor pathway. We were able to generate IECs using this method, and the IECs, the induced endothelial cells, are very similar to true endothelial cells. So these are fibroblasts that have transdifferentiated into endothelial cells upon activation of innate immunity and exposure to exogenous growth factors that are favored by endothelial cells. And you see they can form tubes in matrigel, and they express all the markers that you might hope for. And they also make nitric oxide, and they're just as well as human microvascular endothelial cells. One of the molecules that I've been interested in for years and years is nitric oxide. And so we looked to see what the expression was of eNOS. eNOS is a marker for endothelial cells. And during this process of transdifferentiation, we don't really see eNOS until very late, when we have mature endothelial cells during this transdifferentiation process. So eNOS shows up, endothelial NO synthase shows up at 4 weeks. And at that point, we have mature endothelial cells in the dish. Now, what we saw that was interesting was eNOS shows up much earlier, within a week of the transdifferentiation process. We've actually looked earlier than 6 days now. At 2 days, it's activated, eNOS. No surprise, because activation of innate immunity is known to increase eNOS expression. It is an NF-kappa B dependent. We asked the question, what would happen if we knocked it out? And wild-type MEFs, we get a low yield with our protocol, but we do get cells, endothelial cells, 4.5% yield from this transdifferentiation process that I showed you. But if in the eNOS knockout MEFs, that's almost obliterated. Here's the average data, INOS knockout abrogates transdifferentiation. So we began to look at what INOS is doing. Now, INOS typically is thought to be a cytoplasmic enzyme. But what we observed during the transdifferentiation process is that INOS began to translocate to the nucleus. And I had not known that that could even occur. Looked through the literature. There was actually one report of this in the literature, that INOS can translocate to the nucleus, but no one had described what it's doing there. And what we found out was that when we did a bioinformatic search, 20% of epigenetic modifiers contain a binding sequence for, a consensus sequence for INOS. So we thought, wow, that's impressive. Let's take a look at, some potential epigenetic modifiers that may be nitrosylated. So ring 1 is an important part of the polycomb complex. It has several cysteine molecules, sulfhydryls, that are in a position that could affect the binding of ring 1 to the rest of the polycomb complex. So we looked at that as a candidate. And we found that ring 1 is binding, in fact, to INOS with the IP studies that we did. And furthermore, it's getting nitrosylated. And we did some mass spec studies to show that actually each of the 3 cysteines that are available for nitrosylation are getting nitrosylated during transdifferentiation. And we went on to show that the complex is falling off the chromatin as a result. And this schematic just basically shows the hypothesis. INOS is binding to ring 1, nitrosylating it. The polycomb complex falls off, and that causes a reduction actually in H3K27 trimethylation. The polycomb 1 complex actually is a H2K119 ubiquitination that it's responsible for, but that stabilizes the H3K27 trimethylation. So without this lysine 119 ubiquitination that PRC is responsible for, PRC1 is responsible for, the H3K27 trimethylation of PRC2 is not stable. So that removes a major suppressive influence on chromatin. Here's the idea again. Pattern recognition receptors can detect these pathogen-associated molecular patterns, viruses, microRNA, whatever you're exposing the cell to, activating innate immunity. INOS is one of the mediators modifying the epigenetic modifiers to increase epigenetic plasticity. Here's some data with poly-IC. Oops, sorry about that bar. I don't know why that keeps showing up. Let's do this. Anyway, the idea is that poly-IC increases H3K4 trimethylation, seen here. In the wild-type cells, it reduces H3K27 trimethylation. And you can see in the INOS knockout, those changes in the histone markings are not seen. So here's some new insights, and I don't have enough time to go through the data with you, but this is all new data. We're now moving in vivo, and we're finding the same process occurs in vivo. In the hind limb ischemia model, using lineage tracing, we've been able to see that there is some contribution of fibroblasts, actually a significant contribution of tissue fibroblasts to this, what we call angiogenic transdifferentiation, a transdifferentiation that's occurring in vivo, and that is under the influence of inflammatory signaling and a glycolytic shift that plays a very important role in histone acetylation. So we now have some very nice data showing that a glycolytic shift is involved in transdifferentiation. It's a direct connection of the metabolic program with epigenetics, increased generation of citric acid by the mitochondria is exported to the nucleus, where it's converted to acetyl-CoA for histone acetylation. It's an absolutely critical metabolic shift that is responsible for epigenetic changes. And then some of the work from Xu-Meng, recent work from Xu-Meng, showing, in fact, there's a small amount of transdifferentiation using her cell lineage tracing model. YFP positive endothelial cells basically represent fibroblasts in the ischemic hind limb that have transformed into endothelial cells. It's a small number by comparison to the total YFP positive population, but it's about a third of the entire CD144 population. So it contributes significantly to the revascularization of the ischemic hind limb. And when we block this process, in data I won't be showing you today, when we block the transdifferentiation, innate immune activation of this transformation process, we lose these cells. So innate immune activation is important for this transdifferentiation, and the limbs become more severely necrotic of these animals with the ischemic hind limb model. So in conclusion, we've got this more data to support this model that when a cell is confronted by a challenge, whether it's a pathogen or it's a damage, that activation of innate immunity, in addition to causing a cascade that leads to the production of inflammatory cytokines, causes changes in epigenetic modifiers, changes in their expression. I showed you I showed you the effect on histone markings, and I showed you the effect on DNA accessibility data that we have. There's a lot more data behind that to support the idea that epigenetic plasticity is induced by innate immune activation. So thank you very much for your attention. Thanks to my colleagues who have helped me in this work, and I look forward to hearing what the rest of you have to say today. Thank you so much. In the interest of time, actually, we do have 1 minute for questions, if there are any. Yes?
Thanks for a good talk.
Song Lee.
John, fantastic talk. I have a question. So, in response to vascular injury, people have reported this endothelial cell to mesenchymal transition. So, you are reporting kind of a reverse process. How can this explain what happened in a pathological situation?
Great, beautiful question. Because in the same setting of activation of innate immunity, you can see the reverse, right? You can see fibrosis. And there's good evidence that there's an endothelial to mesenchyme transition that plays a role in that fibrosis, such as cardiac fibrosis in the diabetic. So it's a great question. And I think the answer is the milieu. So you can put the activation of innate immunity puts the cell in a state of plasticity. So it can respond to the environment, so it can change its phenotype. And that change in phenotype can be advantageous or it can be disadvantageous. It depends upon what other factors are acting on the endothelial cell. If we simply add poly-IC to fibroblasts, nothing happens. But we have to add something else. And I mentioned the factors that we use, VEGF and FGF and BMP4, TGF-beta receptor inhibitor. So you have to change the environment so that the cell can respond to that increase in epigenetic plasticity. Great question. I think we have to move on. We can talk more at the coffee break. I'm going to introduce. Oh, okay. I just learned from my co-moderator that Dr. Boley is on his way, not here yet. So we're going to have Dr. Poo, Bill Poo,